Welcome my dear students. Today we explore the amazing building blocks of proteins, that is amino acids. These tiny biomolecules are the building blocks of proteins, and without them life simply cannot exist. We will quickly understand their basic structure, important functions, and how they are classified based on different structural properties. Stay tuned as we make amino acids easy, logical, and exam focused. So the basic structure of the amino acid is here. This is the central carbon atom, and with this carbon there is one hydrogen atom and one amino group, a carboxyl group, and there is a variable R group. Among the different amino acids, the R group is different from each other, but other than the R group, the remaining structure is same in all amino acids. So the functions of amino acids include that they act as the building block of proteins. They forming part of the coenzymes. They can be precursors of biosynthesis of different molecules like hormones, immunoglobulins, etc. Amino acids can be classified based on their polarity, based on their essential features in the body, based on their R group. Now start. So the protein can work as the workhorses of the cell. Protein is the most abundant macromolecule in living cells. They perform diverse biological functions, like they can act as enzymes, transporter, hormones, and structural components. The convert genetic information into action. Protein convert the genetic information which can be found either in DNA or RNA, and it can be converted into action. Protein is made up of 20 different amino acids, and proteins are the polymer of amino acids. That is, amino acids are linked by peptide bonds, that is covalent bond, and each amino acid when added they are called as residue, and then water molecule is lost. Asparagine is the first discovered amino acid, and it was in 1806, and threonine is the last identified amino acid in 1938. Their names reflect the origin. Asparagine is derived from asparagus, glutamate is from wheat gluten, tyrosine is from cheese, that is Greek tyros, and glycine is derived from sweet, that is Greek glykos. Glykos means sweet. So this is the structure of protein, is formed by the bonding of different amino acids through the polypeptide bond, and that is the primary structure of a protein. Subsequently, secondary, tertiary, and quaternary structure is found among the protein structure. So the basic structure of amino acids will start today. All 20 standard amino acids are alpha amino acid because their chiral alpha carbon is attached to the carboxyl group, amino group, hydrogen atom, and an R group. So this carbon is called as the alpha carbon. R group can be varied. It can be small or large. It can be hydrophilic or hydrophobic. It can be neutral or charged. Naming carbons: carbons in the side chain labeled as beta, gamma, delta, epsilon, and outward from alpha carbon. Chirality of amino acid, that is, except glycine all amino acids are chiral, that the four different groups are present, they are attached with the carbon atom. Due to alpha carbon attached to four different groups, amino acids can be exist as L and D forms. In proteins in living organisms, in most cases they are L amino acids, but D amino acids are very rare. They are found in bacterial cell wall and antibiotics. Amino acids general structure: protein is the linear polymer of alpha L alpha amino acid, and they are linked together by covalent peptide bond. That is the general structure of the amino acid. The alpha carbon is attached with the amino group, carboxyl group, hydrogen atom, and variable R group, and this is the numbering of the different carbon atom from the alpha to on outwards. That is, the beta carbon which is attached to the alpha carbon is named as beta. The carbon which is attached to the beta carbon is called as gamma. The carbon attached to the gamma carbon is called as delta, and the carbon attached to the delta carbon is known as epsilon, which is always although attached with an amino group. L alpha amino acid general structure: they can be ionized form at physiological pH, that is L amino acid, that is the NH3 is the protonated form, and it is the deprotonated form, C double O minus. Here this is D amino acid, this is the protonated form. So the alpha carbon from the chiral center, which is optically active, and when it tetrahedral arrangement is there, two stereoisomer can be formed, which are known as enantiomer. So these are the different kinds of amino acid. The non-polar amino acids are glycine, alanine, valine, cysteine, proline, leucine, isoleucine, methionine, tryptophan, phenylalanine. Polar amino acids are serine, threonine, tyrosine, asparagine, and glutamine. And polar charged amino acid, and these are the polar uncharged amino acid. Polar charged amino acids can be two types, positively charged and negatively charged. Polar positively charged amino acids are lysine, histidine, and arginine. Our polar negatively charged amino acids are aspartic acid and glutamic acid. Classification of amino acids: they can be classified based on structure and their chemical nature. So it can, R group can be aliphatic side chain with OH group such as serine, tyrosine, threonine, etc. Side chain with S sulfur, that is found in the sulfhydryl group cysteine, and methionine also is a sulfur containing amino acid. Side chain with acidic group such as aspartic acid, glutamic acid. Side chain with basic groups such as arginine, histidine, lysine. Aromatic amino acids such as tryptophan, tyrosine, phenylalanine. Amino acid is the proline. Based on polarity, amino acids can be hydrophilic or that is polar, or hydrophobic, it is non-polar. Amino acid can be glucogenic and ketogenic, or both glucogenic and ketogenic. Nutritional classification: amino acids can be essential, non-essential, or semi-essential. We'll discuss in detail later. Classification based on structure and chemical nature. Amino acids with aliphatic side chains. There is GAVLI, GAVLI. GAVLI is the glycine, alanine, valine, leucine, isoleucine. They are the amino acids with aliphatic side chain. So, amino acid with hydroxyl group, OH group side chain such as serine, threonine, and tyrosine. Amino acid side chain with sulfur atom, that is sulfhydryl group or mercapto group, where there is SH group is there, and SCH3, that is thioester bond, is found in methionine. When two cysteine amino acid are joined with the help of disulfide bond, from cysteine is formed. Amino acid side chain with acidic group or their amides, acidic amides, aspartic acid থেকে তৈরি হচ্ছে asparagine, and glutamic acid is be converted into glutamine. So the aspartic acid and glutamic acids are the dicarboxylic monoamino acid. Side chain with basic group, basic amino acid, that is dibasic monocarboxylic acid, that is when guanidino group is present in arginine, epsilon amino group is present in lysine, and imidazole group is present in histidine. Aromatic amino acid. Aromatic amino acid are phenylalanine, tyrosine, and tryptophan. Their structure is here. So when it is modified, 7 amino acid proline can be modified. Classification based on polarity. Amino acids can be hydrophobic, that can be non-polar, that is non-polar aliphatic acid are the alanine, valine, leucine, isoleucine, proline, methionine, and aromatic are the phenylalanine and tryptophan. Hydrophilic polar can be negatively charged or positively charged or uncharged. So hydrophilic polar or uncharged molecules are amino acids are serine, threonine, tyrosine, asparagine, glutamine, cysteine, glycine. Hydrophilic polar which are positively charged are lysine, arginine, and histidine, and negatively charged amino acids are aspartic acid and glutamic acid. Classification based on metabolic fate, that is it can be glucogenic, both ketogenic and glucogenic, that is phenylalanine, isoleucine, tryptophan, and tyrosine. Ketogenic amino acids are leucine and glycine, and remaining 14 amino acids such as alanine, aspartate, glycine, methionine are the glucogenic amino acids. So now we know about the uncommon or modified amino acid. There are two major categories which are formed after proteins are synthesized, and secondly directly inserted during protein synthesis such as selenocysteine. For example, 4-hydroxyproline found in collagen and plant cell wall, 5-hydroxylysine found in collagen protein, 6-N-methyl lysine found in myosin protein for muscle contraction, gamma carboxyglutamate found in prothrombin responsible for blood clotting, calcium 2 plus is a calcium binding protein is there, desmosine that is found in elastin, for the proper tissue elasticity. Selenocysteine is the 21st amino acid. It contains selenium instead of sulfur like cysteine. It is derived from the serine amino acid. Directly incorporated during translation, so it is present in limited protein, that is trace but essential protein. Non-protein amino acid. 300 amino acid exist in cells but not used in proteins. Examples of biological significance: ornithine, citrulline, etc. They are important in urea cycle, arginine biosynthesis, etc. This is the 21st amino acid, selenocysteine. The 3-letter code is ACC per sec, and 1-letter code is U. Glutathione, it can act as glutathione peroxidase, deiodinase, glycine reductase, and thioredoxin reductase. So when the selenium group, the sulfidyl group of cysteine is replaced with the selenium group, selenium moiety, it is known as selenocysteine. 22nd amino acid is the pyrrolysine. Pyrrolysine is the 3-letter code is PIL and 1-letter code is O. UAG, normally a stop codon. It is similar to lysine but with an added pyrrolysine ring linked to the lysine side chain. Methyl transferase enzyme form a methane producing bacteria. Thank you my dear students for joining this class. Amino acids may be simple but their role in life is huge. Keep revising the structure, properties and classification to score confidently in your exam. If your concepts of amino acids are strong, then protein structure and metabolism part will become very easier to you in future studies. See you in the next class. Keep learning and stay curious. Bye.